Welcome, everyone. Thank you for joining today. For anyone that is attending this call for the first time, we hold this industry-wide call every month for any company that wants to participate in the cultivated meat value chain. And so the purpose of these calls is really to introduce and welcome new solutions providers into the industry with the end goal of establishing new collaborations that can accelerate R&D and eventually commercialization. So if your group is interested in presenting on a future call, or perhaps you're working with a company that could benefit from being on these calls, please do feel free to put them in touch with me directly. So some housekeeping items on our end. There's still time to complete our bioprocessing survey that we have out right now. Thank you in advance to the 22 companies that have completed it thus far. So if you're a cultivated meat company, especially a manufacturer that has a functional facility, or you're planning on building a facility, please do prioritize filling out that survey in the coming days if possible, and I'll drop the link in the chat just for reference. If you're like developing scaffolding or media or those sorts of things, don't worry about this one. It's not applicable to you. So hopefully people saw the news with Good Meat receiving the green light from the FDA on Tuesday. And so just wanted to share that we do have an explainer on our website of the US regulatory pathway in case you're receiving questions and would like to point people to an easy sort of overview and explainer. I'll drop that link in the chat too, so you have that reference handy. And then finally, we'll be hosting a webinar on April 12th that will overview the findings from our recent updated life cycle analysis of cultivated meat. This looks at the environmental impact of cultivated meat using data from over 15 different companies and that participated in this study with GFI and was performed by CE Delft. I dropped the registration link in the chat, and I also wanted to let folks know that I posted a summary slide deck publicly. So I'll drop that link in the chat, and you can feel free to use these slides in your own presentations when talking about cultivated meat, if you'd like. It's free to download, free to use, etc. Please feel free to go through that and use however you'd like. All right, so with that said, today's speakers, I'm really pleased to welcome Daniel Benjamin from Metalytics, who will be speaking today about how metabolic flux analysis can improve your bioprocess. And then we'll turn it over to Simon Hubbard from Upstream Applied Science, who will be sharing some of his work on developing open source techno-economic analysis workflows. As always, feel free to drop questions in the chat throughout, and we will get to them at the end. So Daniel, I'll turn it over to you.
Okay, thanks so much, Elliot. Let me share my screen here. Okay, so again, thank you all so much for coming. Thanks, Elliot and Renee, for organizing this. I really appreciate it. And so today I want to talk about how at Metalytics we can use metabolic flux analysis to improve the productivity of cultivated meat. So as you guys are all aware, cultivated meat involves the production of edible biomass via the in vitro culture of precursor cells. And again, as you guys know, this is both an environmentally friendly and ethical alternative to traditional animal farming. However, if this is going to be a viable long-term alternative to conventional meat, cultivated meat must be both cost-effective and widely accepted by the consumer. And one of the major hurdles faced by the cultivated meat sector is the ability to rapidly and cost-effectively expand these cells in culture. And when we talk about the ability of cells to expand or proliferate or grow, what we're really dealing with here is a question of cell metabolism. So fundamentally, if we want to understand how to optimize this process of cell expansion, we have to understand cell metabolism. So first of all, what is cell metabolism? So cell metabolism consists of, number one, catabolic pathways, which enable a cell to take in nutrients and break down those nutrients into both energy and the building blocks, and anabolic pathways, which combine the energy and the building blocks to allow a cell to produce macromolecules that eventually enable that cell to grow and proliferate. And so again, if we want to optimize the process of cell growth, we have to gain an intimate understanding of the metabolism that underlies that growth. However, the challenge here is that metabolism is highly complex. There are thousands of different biochemical reactions, and there are thousands of different possible permutations of these biochemical reactions. And so this can often feel like kind of a black box. And so how do we visualize what's in the black box? How do we visualize cellular metabolism? And at Metalytics, we take an approach called metabolic flux analysis using our platform called Core MFA. So what is metabolic flux analysis? Well, the word flux literally means the rate of conversion of one thing to another. And so metabolic flux is the rate of conversion of one metabolite to another in metabolic space. And the beauty of metabolic flux analysis is that not only does it tell you how fast metabolism is unfolding, but it tells you exactly the direction by which metabolism is unfolding in a cell. And so we say it tells you both the rate and the route of metabolite flow. And metabolic flux analysis often gets unfortunately confused with metabolomics. Metabolomics is very simply measuring the abundance of metabolites at a snapshot in time, but tells you nothing about the speed of metabolism or exactly the direction by which metabolism is unfolding in a cell. I think one useful analogy here is to think about metabolic flux analysis as sort of like Google Maps or the Waze app. And so not only does it tell you where all the roads are, but it also tells you where the traffic bottlenecks are and how you can most efficiently move from point A to point B in space. Metabolomics would be akin to me just giving you a picture of cars on a road, and you wouldn't know how fast they're going or where they're going. So how do we perform metabolic flux analysis? In order to perform MFA, two inputs are required. The first is something called a metabolic model, which I'll explain in a moment. And the second are two pieces of experimental data. And so let me quickly walk you through exactly what these are. Let's start with the metabolic model. So the metabolic model consists of two parts. Number one, it consists of all of the biochemical reactions for the metabolic pathways of interest in the cell. And so for this, every single carbon needs to be mapped from every substrate to every product. And in addition to that, thermodynamic information also needs to be included here. Information like, is the reaction reversible or irreversible? In addition to the list of all of the metabolic reactions, the metabolic model also requires a biomass equation. And you can think about this sort of like the demand function of a cell. So when a cell takes up nutrients, where they need it to go to make another cell, where they need it to go inside the cell. And for this, we need to measure the macromolecular composition of a cell type of interest in order to construct this specific biomass equation. And so by combining all the reactions in the cell with the biomass equation, we can construct a metabolic model for any given cell type of interest. And for example, using this approach, Metalytics constructed a novel metabolic model for a very unique species of cultivated animal cell, using a eukaryotic literature-based model as a base, combining that with measured macromolecular data. Again, we were able to construct this novel metabolic model. In addition to the model, we also require two pieces of experimental data in order to perform MFA. The first piece of data is called external rate data, and the second is called isotopic labeling data. Start with the first one. So if we want to uncover how metabolism is unfolding in a cell, the first thing we need to know is how fast things are entering or leaving a cell. And so, and this is called the external rate data. And so to acquire this, we have to measure the abundance of metabolites in the extracellular media over time. And we can use that information to back calculate exactly the rate by which they entered or left the cell. But again, just because you know how fast something entered or left the cell tells you nothing about the direction by which it moved once it was in the cell. And for that, we need isotopic labeling. So how does it work? Well, I think one useful analogy is to think about a car entering or leaving a city. And just because you know how fast the car entered and left the city tells you nothing about the direction by which it moved when it was in the city, how it traversed from one point to another. But what if we could paint the tires with some red paint and then track where that paint was left behind on the road? And so, sort of analogously, isotopic labeling can track the fate of cellular carbons through cellular metabolism. And the way this works, the way MFA studies are carried out, is by feeding cells with a non-radioactive isotopically labeled substrate, generally 13C glucose, and using mass spec to measure the incorporation of this isotope into downstream cellular metabolites. And so by combining the metabolic model with both external rate data and isotopic labeling data, we can elucidate the metabolic fluxes for all of the reactions of interest in our metabolic model. But you're probably wondering, how can we use this kind of data? How can we use this flux data to optimize the production of cultivated meat? And I want to tell you about two projects that Metalytics worked on with two different companies, where we used MFA to improve cultivated meat for these companies. So the first involves us deploying MFA in a very unique animal species to elucidate the flux, the metabolic flux in this species. And by doing so, we were able to identify a very critical metabolic bottleneck in these cells. And the second project, the second project with a different company in a different animal cell type, involved us deploying MFA to improve the expansion of these cultivated cells to help work towards a more defined media. So we'll start with number one. So we were working with a company that was working with a very unique cultivated animal cell type, and they had a specific problem, which is that their cells were unable to continue to grow after a certain amount of time in culture. And the question fundamentally that we asked is this reduced growth rate the result of specific metabolic bottlenecks and/or nutrient limitations? And how can we use our core MFA platform to overcome this problem? So we could ask the question, what are the metabolic bottlenecks that limit the efficient growth of these animal cells? And so we were able to elucidate the metabolic flux in this very unique species of animal cell. And here what I'm showing you is the pathway glycolysis. And so the pathway starts with glucose and ends with a couple molecules of pyruvate. And this pyruvate can then go on into the TCA cycle to produce ATP energy, or can be essentially expelled from the cell via the waste product lactate. And what you're seeing here are the rates in nanomole per million cell per hour of various enzymatically catalyzed reactions of glycolysis. And what we observed here, quite interestingly, was that the rate of glycolytic flux in these animal cells was significantly slower than what we had seen in other types of animal cells. Yet the rate of lactate waste production was significantly higher. Again, these cells seemingly were not efficiently moving carbons towards the production of energy, but instead, a lot of those carbons were being wasted from the cell, being secreted as lactate. And so the next question is, well, how can we use core MFA to overcome this problem and provide a solution to this problem? And so using MFA, in addition to identifying these metabolic fluxes that you see here, we also identified the potential for the utilization of galactose in this unique species of animal cell. And galactose is very interesting because it's known that galactose metabolism is more oxidatively efficient. It's more energy efficient than is the metabolism of glucose, and can be used to supplement with glucose to enhance energy efficiency and oxidative metabolism. And so perhaps this data revealed to us that we could supplement the media with the metabolite galactose to improve the metabolic efficiency and increase the energy production in this species of cultivated animal cell. And so the second project we worked on, I'll describe below. And so in this project, we were working with a company that was looking to move towards a chemically defined media, away from the use of serum. And the problem they were having was that the ability of these animal cells to grow in culture in serum-free conditions was limited after a certain amount of time. And so we asked, again, the fundamental question, is this reduced growth rate the result of specific metabolic bottlenecks and nutrient limitations? And again, the question is, how can we use metabolic flux analysis to identify and to overcome this problem? And so when we talk about the cellular requirements needed to grow and expand, we're really dealing with, as I mentioned before, two things. ATP energy. A cell needs ATP energy, and it needs the building blocks. And so we can interrogate these two possibilities separately. We can ask, perhaps ATP energy might be limiting for the ability of these cells to grow, or perhaps it might be one or more of the building blocks that might be limiting in the ability of these cells to expand in serum-free conditions. And so first we can look at ATP energy. And so using our metabolic flux analysis, we were able to calculate the rate of ATP production in these cells. And what we identified was that although, as I mentioned earlier, the cells in serum-free conditions were growing at a slower rate, the biomass flux was less, we noticed that the rate of ATP production in nanomole per million cell per hour here was no less, in fact, was slightly more than what we were seeing in serum containing conditions. Again, suggesting to us that ATP was likely not the limitation here. So what about the building blocks? Well, using MFA, as I'll show you in a moment, we were able to identify whether or not specific amino acids might be limiting for the production of biomass to enable a cell to grow. So how do we do that? Well, at the high level, the way it works is that when a cell takes up amino acids, it can do one of two things with them. It can use them to produce biomass to allow a cell to grow and proliferate, or it can use them, it can catabolize them, burn them for energy. And the way the metabolic model works is that all the amino acids are first prioritized for biomass, and then anything that is left over is catabolized for energy. And essentially what you can do is you can use this technique to identify whether or not an amino acid is limiting for biomass production if its catabolic flux rate is 0. And so we asked the question, were there any amino acids for which the catabolic flux rate was indeed 0? And we identified a specific aromatic amino acid that had a 0 catabolic flux rate in serum-free conditions during late growth. Again, suggesting very likely that this aromatic amino acid might be limiting for the growth of these cells. Going further, we asked what might be causing this limitation. And we uncovered the fact that, well, not only is this amino acid limiting, but the rate at which these cells are able to uptake this amino acid is significantly reduced during late growth. And so how do we overcome the bottleneck? How do we overcome this potential limitation of this specific aromatic amino acid? And so one way to do this is to supplement this chemically defined media with a dipeptide containing the limiting amino acid, in this case, containing this aromatic amino acid. Why a dipeptide? Well, we know that if there's some problem with uptake of an amino acid, the addition of the amino acid as a form of a dipeptide can overcome certain limiting kinetics. And so hopefully I've showed you guys today how we've used metabolic flux analysis to elucidate the metabolic flux of 2 distinct animal cell species for cultivated meat production. And specifically, I showed you one example using one specific species where we identified a specific inefficiency in energy metabolism. And I showed you a separate example in a different species where we identified a specific amino acid limitation during serum-free growth. And so what lies ahead? Well, Metalytics is very excited to continue to work in the cultivated meat space, not only to include additional animal cell species, but also to look at the cell differentiation phase, because the ability of cells to differentiate is also fundamentally a metabolic question, which MFA can be used to help understand and to answer and to identify bottlenecks in this process. And so with that, I'd be happy to take any questions. I'll just leave you with the contact slide. If you're interested in any of this technology or would like to reach out, feel free to reach out to me or to anyone else on our leadership team. And so thank you guys so much.
Awesome. Thank you, Daniel. These are great case study examples. And it's great to see you providing value for some of these companies already. So anyone have questions, we can come off mute or drop them in the chat. Thank you, Daniel, for presenting, and we'll pass it over to Simon now.
Thanks, Elliot. Hi, everyone. Let me just share my screen. Okay, so my talk today is around development of open source techno-economic analysis workflows, particularly for applications in the cultivated meat space. So this is an ongoing piece of work that's funded by the Good Food Institute. I was successful in the RFP last year, so this is work in progress. And one of the key parts of this activity is to do this development as openly as possible. So thanks to Elliot and Renee for the opportunity to share this with everybody today. So in terms of the intent of the project, it's to build on existing techno-economic analysis workflows in a couple of ways. So there's an aspect of technical development, and so that's really concentrating on the techno part rather than the economic part. And to try and remove some of the assumptions of existing models, combine with other analysis methods that give you more detailed descriptions of the technical performance of the cell lines and the bioreactor operation, and then assess scaling and predict production scale yield, and use that capability to be able to optimize and explore bioreactor designs in terms of optimizing and maximizing yield. And then, as I said, the intent to do that from an open perspective and to make this workflow as usable as possible. So from a usability point of view, the idea here is to try and maximize the amount of flexibility we have with inputs into the workflow. So some of the inputs are indeed quite sensitive for a number of companies and organizations in this space. So the idea is to be able to give people as much flexibility in providing inputs that might contain sensitive data and be able to construct the workflow around that intent. So in terms of the presentation today, there's a few different things I'm going to try and put together. I'll talk through techno-economic analysis, a little bit of background, cover a little bit of open workflow development, and then I'll touch on genome scale metabolic models, which touches very closely to the presentation we've just seen from Daniel, and then add in a little bit of computational fluid dynamics. The idea there is to then give you a sense of how we can bring those things together to have a tool set and a workflow that gives us a yield prediction to help us with scale up and bioreactor design to hopefully get us to the situation where we've got cultured meat available on the shelves as soon as possible. I'm not going to do a particular deep dive into any of these things technically. So the metabolic models and the computational fluid dynamics are more focused around how we can use those tools to build on the framework of techno-economic analysis rather than doing a particular technical dive into any of those particular aspects. Okay, so in terms of techno-economic analysis, or TEA, they're high-level technical models combining aspects of bioreactor performance, cell growth and yield, coupled with economic cost models from a production line perspective. There's a number that have been published in this space, and one that's particularly good from a technical detail point of view, and people may be quite familiar with, is David Humbard's publication from 2021. So I've just grabbed a couple of images from that publication, which is nicely open access. There's a reference at the bottom there and a link to go and have a look if you want, and the Creative Commons open source license. So the idea here is then to kind of summarize what happens in a TEA. So you come up with your production architecture. So there's a couple of examples here. In the top case, we've got a fed batch production scenario, in the bottom perfusion, and then to use models from cell growth, cell behavior, bioreactor performance to come up with two outputs. So the graph in the top right here is a cell growth output in terms of the achievable yield in terms of cell density against the number of constraints that you might have around dissolved oxygen, cell carbon dioxide, mixing performance, and metabolic products, and to look at how those things scale with bioreactor volume. And then the graph in the bottom right is to do a similar type of activity, but from an economic perspective as opposed to a technical perspective, so that you can come up with a cost per kilogram of your cultivated meat products that would result from these production lines. So as I said, the presentation today is going to focus entirely on the top right type of analysis, such that you can have more detailed models to give you more specific predictions of cell density and yield perspective, and then you can combine those with existing economic models from a cost per kilogram output point of view. Okay, so in terms of the details in some of these technical aspects, there's four main parts here, and this is well described in that published work from David Humbard, and I'll just go through these to give a little bit of a perspective. So you've got a cell reaction and a growth model. So from published work, you've got a stoichiometric reaction in terms of uptake and production of different metabolites, and a growth rate. So previously, this has been using Chinese hamster ovary cell data. It's the usual case of lots of data available for those cell lines, and some consideration of how close they might be in terms of behavior to cultivated meat cell lines. And the challenge of getting particular information for particular cultivated cell lines is one that drives into trying to model these sorts of things. And then there's some constraints around that growth. So limits for the concentrations of dissolved carbon dioxide, ammonia, lactate, and some description around the hydrodynamic stress that the cell will experience in the bioreactor, such that you can combine these with the cell reaction model, which has a particular growth rate that you say the cells will continue to grow at until any of these constraints are hit in terms of the experience of the cells in the bioreactor, such that growth stops under those scenarios. So that's a cell perspective. The bioreactor perspective, you have a number of different models that you can use to describe bioreactor performance. They're a mix of first principle models, assumed form models, empirical models, and there's a handful here to give you a sense. So you've got models that describe the concentration difference of dissolved oxygen between your gas phase in your bubbles and aeration and in your media. Got a model for the mass transfer effectiveness of dissolved oxygen from the gas into the media and for carbon dioxide in the opposite direction. Models for the mixing time of your bioreactor and models that describe some characteristics that are related to the hydrodynamic stress that the cells will experience. So there's quite a lot going on in those equations. I wasn't intending to dig into too many details, but they cover things like the power from your agitation and your impellers, the volume of your media, dimensions of your impeller diameters, your tank diameters, liquid height, and so forth. And then in a similar way to the cell situation, where you've got some constraints, you've also got some constraints on your bioreactor performance, with things like the mixing time needing to be shorter than the mass transfer time, such that your oxygen, once it's moved from your gas bubbles into the media, is quickly mixed and uniformly and well distributed throughout the rest of your bioreactor at a rate that's faster than it comes from the gas bubbles, to make sure that you're trying to keep everything as well mixed as possible. And then you've got other things such as the mass transfer of oxygen from your bubbles into your media needs to be happening at a faster rate than the cells are taking it up from your cell reaction at the top here. So when you put all of those things together, these are the sorts of outputs you can get from the model. So this is an example similar to the published work, where you've got some initial conditions in your 20,000 litre stirred aerated bioreactor, and you've got a 48-hour growth period, some initial cell density characteristics, and a constant growth rate over a period of time, which then stops after 48 hours because you've got a buildup of ammonia that's hit the 5 millimoles per litre limit that you've specified over here as part of your cell growth constraints. So you then have the ability to change your initial conditions, the duration, and look at where you may run into different constraints in terms of metabolites or dissolved carbon dioxide or mixing constraints against that framework. The other sorts of things you can do with that is then reframe that in a different way, where you can now look at how that yield, achievable yield limited by all of those different constraints, is varying according to your bioreactor operational parameters. So two key parameters here would be your impeller agitation, RPM on the x-axis, and the rate at which you're injecting gas into your media. So you've got an aeration velocity here. And you can see in this little landscape, you've got a very flat area where you've hit this 7 grams per litre wet limit, and that was due to the ammonia limit from the previous slide. So within this parameter space of agitation and aeration, you can't beat your 7 grams per litre yield limit as a result of the ammonia constraint you saw from the previous slide. But with that in mind, you can still then use this approach to start to dig into some other constraints to see what your yield limit might be if you weren't limited by ammonia, for example, or some other metabolite concentrations. So you can break the same picture down in terms of agitation and aeration for particular constraints that we've just been talking about. So from an oxygen mass transfer perspective, you can see at high agitation and high aeration, you can hit 40 grams per litre up in the top right corner here. Slightly different and simpler pictures in terms of cell stress and in terms of superficial velocity constraints, carbon dioxide on the bottom, and mixing here. And so the other nice thing you can do is then start to understand how you have this landscape for your overall yield, which is limited by ammonia concentrations. But you can see that this shape in the bottom left is driven by your oxygen mass transfer. This hard cutoff on the right is driven by your cell stress, and this cutoff on the top is driven by your aeration velocity. So you start to get to a sense of where these constraints come from in terms of very specific drivers in terms of a bioreactor constraint or a cell growth constraint. So those things that I've just described have been put together in a Python module that's open access in a GitHub repository on the shared address there. So that does the two things. There's a couple of examples you can see. There's a basic yield prediction, which gives you the kind of graphical output from the first case, where you've just got a graph showing what happens as a function of time. And then you've got this rather simple brute force optimization approach, which allows you to look at how things vary as a function of bioreactor operational characteristics. So that's really a model that captures existing capability. So the next question then is, how do we build on that capability to get some more detail into the models and remove some of the assumptions? So one of the assumptions that's in the cell reaction and growth model is that this stoichiometric reaction has got coefficients that are constant with time, and they're not sensitive to the media concentrations. So this model was built on the back of a lot of research into CHO cell anabolic and catabolic reactions and built accordingly, but it still assumes everything continues at constant rates with a function of time. So one way that you can start to expand on that is to use genome scale metabolic models. So this touches on the presentation that Daniel's just provided, and he's given a much more detailed and involved presentation than I could on the background for that. But in the context here, one thing that you can do is you can use existing functionality in terms of Python modules. So there's a Python module called CobraPy, which allows you to do the sort of metabolic flux analysis that Daniel was describing, and also allows you to download existing metabolic models that other people have built. Again, this is a similar situation to what we may be familiar with, in the sense that Chinese hamster ovary cell models are nicely available, not necessarily as relevant for cultivated meat cells as we might like. But in terms of a capability here, where you can then download existing or pre-developed metabolic models, or build metabolic models, particularly for this type of application, you can follow this sort of progress, where once you have a model that you've built or someone else has constructed and you've downloaded and using in this library, you can then optimize that metabolic flux pathway. So that really maximizes the growth rate, given some constraints around the rate at which fluxes are the fluxes of uptake and secretion in terms of metabolite products and amino acids. So here, what you can see is, for this example, you can get a range of uptake fluxes and a range of secretion fluxes for the same type of amino acids and products that we were talking about previously. The nice thing here, and the thing that's important for this type of workflow, is then to be able to provide bounds to some of these fluxes. So from the perspective here of a situation where there might not be as much oxygen availability in the media, you can bound the rate at which the oxygen uptake flux occurs at, and rerun the model. So then you get a sense of a situation now where you've bounded the rate at which oxygen's being absorbed at, and this has knock-on effects for some of the other uptake fluxes. So you can see glucose is changed slightly here. The rate of secretion of carbon dioxide is changed as a result. So that's the nice way that you can then use these type of metabolic models to allow the overall techno-economic analysis workflow to capture time-varying behaviors as a function of media concentrations. The thing that you need to do here is to combine this approach with a kinetic model that allows you to translate the concentrations in your media to the fluxes, the uptake fluxes that the cells will be operating at. So there's an extra piece of combining this type of metabolic model with kinetic models to allow you to convert concentrations in the media to fluxes that the cell experiences. So another aspect then is to look at bioreactor performance models. So all of the ones that I showed earlier were built on existing data for bioreactors of different scales and in different operating conditions. So the challenge there is, when you've got good data for a production scale bioreactor or a lab scale bioreactor, to then have something that's translatable across different scales. So we've got models for, here's a good example. So your mixing time that's a model that was built for large-scale bioreactors, and it's not obvious if that's translatable to lower scales. So how do you combine that need with this workflow to account for different performance relationships at different scales? So the idea here is to allow the workflow to take in bespoke models that people can develop according to their own experiences, either at a lab scale or moving towards production scales. And you can write your own equation form. So there's an example here. This is the mixing time model that's used in previous data. And so you've got some coefficient values that you could tune your model to as a function of power, volume, different dimensions, and so forth. And so the idea here is to be able to allow people to specify their own models in this form, or to use data fitting approaches. So Gaussian processes is a really nice way of fitting relationships and models to existing data sets. So if you've got a bunch of data that you can sample from your benchtop process, you could build a Gaussian process model and use that as an input into the techno-economic analysis. And the nice thing here is you can combine both experimental and computational data, which helps from a scaling perspective. So you could use a situation where you have experimental and validated computational data at the lab scale, and then building on that validated computational data to have a predictive computational model at production scale before you've been able to build and assess them. So a little example over here on the right is if you wanted to do this type of activity using computational fluid dynamics to give yourself a characterization of your mixing time relationship, you can run a CFD model here of a standard bioreactor, input a little bit of dye in the top left-hand corner and observe how that dye is mixed within the bioreactor, and you'd get a certain output, a probe location that you could specify somewhere else within the media. And that allows you to characterize the mixing time, and then you can use that computational model to look at different impeller geometries, different RPMs, different other geometrical characteristics of the bioreactor, and use that at different scales according to what's going on in your lab, or as you're looking to increase scale accordingly. So another part that you can build on in terms of these bioreactor performance models is to look at hydrodynamic stress. So this is a particularly interesting and quite a challenging area to have a think about. So a lot of the existing work talks around hydrodynamic stress on cells being characterized by a length scale of the turbulent motion in the bioreactor. So the idea here is that there's a scale of energy transfer and turbulent flows where you've got large scales that are created by impeller movements, and then the energy transfers from those large scales to smaller scales where it's dissipated to heat. And so one way you can do the maths around that energy transfer is to look at a Kolmogorov length scale, which is the smallest scale of turbulent motion at which energy is dissipated to heat. And the idea here is that if that smallest scale is larger than the scale of your cells, so larger than your cell diameter, then the cells don't experience that energy dissipation and don't experience hydrodynamic stress, and so don't have any impacts or impediments on their growth as a result. That's a nice way of thinking about that comparison. It's a nice equivalence in terms of length scales. You've got a length scale of a flow and you've got a length scale of your cell, and you have a situation where, as shown in the cell growth constraint up here, where you say that your length scale of your turbulent flow needs to be larger than your cell diameter, an example being 20 microns. Other ways of thinking about it are not so specific around this length scale and just say, actually, for a particular cell line, the energy dissipation rate can't be particularly high. So there's a limit on what this energy dissipation rate is, and that limit is scale independent, and we just need to keep that limit, that maximum rate at which energy is dissipated to heat, below some level, and that level is specific to a given cell line. So some of the challenges around this are how you measure your dissipation rates and how you relate that to the cell experience. So this picture here at the moment on the screen is from a computational fluid dynamics model where you've looked at the time averaged eddy dissipation rate in the region around the impeller. So this is the area that you would focus on. The turbulence is strongest in this region. And so measurements tend to focus on this time averaged dissipation rate as a result of the limitations of measurement approaches where you've got laser techniques where you try and measure things over time and average them in particular locations. And so this is the sort of profile that you will see from computational studies and from experimental studies, where as these impeller blades on this Rushton turbine move around, you'll see that you've got particular dissipation rates close to the impellers. And these are the dissipation rates that people think of at the moment in terms of what the cell will experience. However, you can look at the situation in computational fluid dynamics where you don't look at the time averaged dissipation rate. You can look at the instantaneous dissipation rate. So here, this is essentially the data that has been time averaged to give you the picture on the top. But you can see here the instantaneous rates peak at values up to somewhere around 7 times the time average rate. So the thought process here is then, in terms of cells that are moving around in the media, they won't experience the time average rate, they will experience these instantaneous rates. So a way that you could use that to help characterize this situation in a bit more detail is then to use your computational fluid dynamics model with particles that represent the cell trajectory as they move around in your bioreactor, and then you can track their experience history over time, so that you can generate graphs like you can see here, where you've got over a 60-second cell experience, you've got energy dissipation rates that the cell experiences, and you can see a couple of situations here where it's likely moved close to the impellers within the bioreactor. So this takes existing ideas around cell lifelines, which have been published for fermentation applications, and applies it in a slightly different way to talk about hydrodynamic stress on cells from a cultivated meat perspective. So that's a nice way that you can start to use computational fluid dynamics specifically to expand on some of the modeling approaches that you would use in your TEA. Some of the challenges there are then correlating these types of things to cell growth experiences, but it's a nicer way that allows you to have a maybe deeper picture of what you think the cell's experiencing from an energy dissipation perspective, compared to some of the current approaches that are used in these models. Okay, so that was quite a lot of areas to talk through in terms of different aspects of the TEA workflow. So what's next? So there's a continuing development exercise to incorporate Chinese hamster ovary cell genome scale metabolic models for the cell reaction modeling aspect, and then to build the capability to have some of these bioreactor specific performance models into the workflow. So that's the development activity from a workflow perspective. The next part is then to use that workflow to take an example scaled production line, talking about 50 to 2,000, 10,000 litre stirred tank bioreactors, to look at yield performance and scalability across that range, and then to use that framework to do some optimization of bioreactor performance in terms of optimizing for yield and scaling across those different volumes. And then secondary to that is to then model alternate bioreactor architectures to try and then expand our understanding of other approaches, such as to kind of go past the stirred tank and look at bubble columns, maybe some fixed fluidized bed type approaches, to look at the performance of different bioreactor architectures in the same context, using the same set of constraints and the same set of models. And a stretch target, I've shaded it a little bit at the bottom. One thing that would be really useful here is to then do a stronger coupling between these metabolic models and the computational fluid dynamics models, to then start to get an understanding of the effects of the spatial heterogeneity of dissolved oxygen, concentrations of amino acids and metabolic products in the bioreactor when you get to large scales, so that you can then start to understand some of the performance effects from that spatial variability. Everything that I've talked about so far in the context of the TEA models assumes that you've got everything is perfectly mixed and you don't have any spatial variability at your larger scale. So there's a little piece of work there to try and combine those two things together as part of the later stages of this project. So in terms of everything that I've talked through there, thank you very much for your time and the attention. I would love to talk to people around general feedback on this approach for building these type of models and expanding them in the sorts of ways that I've described. I'd love to talk more about cell growth constraints. So the values that I shared earlier are from published data. A few people that I've spoken to have pointed out that that's quite a difficult area to capture, and it's not easy to understand what those limits are. So in terms of the workflow here, the workflow allows you to input your own limits, but in terms of development and validation activities, it would be great to talk to people around those type of things. Obviously, cultivated meat, metabolic models and kinetic models from the cell reaction perspective. I'd love to be able to move away from using Chinese hamster ovary cell models and to use models that are more representative of cultivated meat cell lines, and anything around bioreactor characterization and cell stress model and validation in terms of the last activity. Be super to get some input and have some conversations around those things. My email address is there on the bottom for anyone who'd like to pick up on any of those things, and welcome questions, thoughts and comments on any of those topics right now.
Awesome. Thanks so much, Simon. I liked every bit of that presentation, very clear, and really like where these things are headed. We have time for questions if people want to come off mute or drop them in the chat. Well, I have to say this is the dream from our perspective, you know, having all these sort of open data feed into these models that people can plug into and just have that out there is going to be so valuable. But of course, it needs to be done with, you know, coincident with some of the industry and that are actually using these cell lines and growing the cells in these bioreactors. So I do hope folks will engage with Simon and the Metalytics team to you know, improve upon this. And I think this will have a really big impact in the long term for the industry. So thank you to both presenters today, and we'll see you here next month.
Thanks, Elliot. Thanks, everyone.